Active immune systems are immune systems where the organism produces their own antibodies to fight off an antigen. In this video, we will be focusing on two examples of active immune systems, humoral and cell-mediated system. The first one is humoral. Humoral is also known as antibody-mediated immunity. The pathogen tries to enter the body by going through the skin, which are nonspecific cells, but it is unable to. This is because nonspecific cells are the first line of defense against pathogens and it is successful in fulfilling its function. However, the pathogens have another way in, through the mouth. In the body, the macrophage, a phagocyte, roams around trying to engulf any pathogens it finds. The macrophage engulfs the pathogen and digests it partially. It does not digest the antigens on the pathogens and instead replaces them on the MHC2 of the macrophage. Before we continue, what are macrophages? Like skin cells, they are nonspecific cells. Their function is, like stated before, to engulf pathogens since they are pathocytic cells. And another function is to release cytokines, which I will soon get in depth into. With the antigen of the pathogen connected with the MHC2 of the macrophage becomes an antigen-presenting cell. This antigen-presenting cell will then bind to the helper T cell receptors and release cytokines, also known as interleukin-1, to the helper T cells. Wait, so what are helper T cells? Well, they are one of the most important cells in adaptive immunity because they help activate B cells to secrete antibodies and macrophages to destroy ingested microbes and help activate cytotoxic T cells to kill infected target cells. But first, we will look into how it helps to activate B cells. These cytokines are signaling molecules that go through the signal transduction pathway to activate other cells, which in this case is the helper T cells. This is an example of juxtacrine signaling, which is when a cell and another cell communicate with one another from contact. With the helper T cells activated, interleukin-2 is produced in the helper T cell and released to the daughter helper T cells that were produced from mitosis. This is an example of paracrine signaling. This is when a cell releases its signaling molecules and it is received by cells that are around it but are not touching the cell. B cells also engulf the same antigen type and become an antigen-presenting cell like the macrophage. However, a few B cells would not be enough to get rid of all the pathogens that enters the body. This problem can be solved with one of the B cells becoming an antigen-presenting cell and connecting its MHC2 protein that is attached to the antigen with the receptor on the helper T cell, allowing the helper T cell to release cytokines. These cytokines will then go through the signal transduction pathway in order to help activate the B cells to divide, differentiating into cells known as plasma cells. These plasma cells are larger than normal B cells because they have rough ER inside of them that are responsible for the secretion of antibodies. These antibodies are identical and specific to the antigen that was present on the surface of the B cell and macrophage. Other B cells that were also divided that did not become plasma cells eventually become memory cells, cells that will remember the specific antigen in the pathogen the macrophage and B cells encounter so that when they attack in the body the next time, our bodies would be immune to it. That is all about humoral. Now we will get into cell-mediated. Similar to the humoral response, pathogen enters the body through any opening such as eyes, nose, mouth, and ears. An antigen-presenting cell, APC, encounters a pathogen and engulfs it. The lysosomes in the APC breaks apart the pathogen and produces antigens. Then a MHC class II protein combines with the processed antigen. The combined antigen and protein go to the surface of the APC and are now attached to the cell membrane. A helper T cell recognizes the antigen on the APC and binds to the complex. This helper T cell is now activated. The activated helper T cell releases signals that are picked up by cytotoxic T cells. Some cells, like cytotoxic T cells, are called specific defense systems because they have specific receptors with viral antigens that attach to a virus-infected cell. The T cell then initiates apoptosis of the infected cell. This would not be able to happen with any other kinds of cells because the shape of the receptor can only fit with a specific structure. The cytotoxic T cells come to the site of infection and bind to an infected cell. This binding causes the T cell to release perforin, a chemical that punctures the cell membrane. Once the membrane is punctured, the infected cell bursts and dies. When the threat is dealt with, regulatory T cells come over and deactivate the cytotoxic T cells. Memory T cells remain behind in case the same pathogen attacks again. The next time it comes, the cells will be prepared.